So this was really a good start into the morning. Now comes something also very exciting. So Professor Charles Raison is going to present the study data of their phase 2. And that's funny. That's funny. I have the bio of Max Wolf to Charles. So Charles is a professor at the Medicine University and also head of clinical research at Usona. So welcome. And somehow, Charles managed to get the publication of the data certified or yesterday, correct?
Yeah,
last night.
We planned it out so it could be shown for the first time in the world today. So you folks are actually hearing these data for the first time anywhere in the world, which is great. Now, how do I make the slides move? Is it a telepathy thing or is there's some, oh, there it is. I thought it was part of the, well, hello, everybody. I'm Chuck Raison. I am the Director of Clinical and Translational Research for Usona Institute, and I am going to show you data that just came out last night of what I think is really the largest study in major depressive disorder writ large, not treatment-resistant depression, but I do want to set a context. So here we go. Let me start by saying who Usona is. Usona is really a fascinating entity. It is a nonprofit medical research organization founded by Bill Linton, who's sitting in the second row there, who was a visionary in this space. He had a friend and a colleague and a neighbor who was dying painfully of cancer, and the woman was devastated, and she was manifesting a lot of depression and anxiety, and it was distressing her family, as so often happens. And Bill managed to get her into one of the first Hopkins trials. I'll show you a slide in a second. She had psilocybin and had this profoundly transformative experience, and he was like, wow, I want to dedicate my wealth and my passion and my vision to trying to bring psilocybin available to the market as an FDA-approved medication. As we were talking last night, he said, how difficult can that be? And of course, the answer is it can be very difficult. It's been a fascinating adventure. So this is very unusual to basically be a nonprofit pharmaceutical company developing a GMP quality agent, not for a little bitty condition, but for a condition that afflicts millions and millions of people around the world, and that by all measures appears to be worsening, at least in the United States. So I mentioned this trial, and it's a small world. Matt Johnson, of course, with Roland Griffiths, were so visionary in their pioneering work of transitioning the early studies of psilocybin in healthy volunteers into a clinical population. And the clinical population that they chose and that the crew at NYU simultaneously chose were people that were struggling with a potentially life-threatening cancer and who developed depression and anxiety around that situation. And I love to show this slide because I think that the headline, this headline actually, I think, set us up for a lot of challenges because a dose of hallucinogen, a dose from a magic mushroom, and then lasting peace, suggesting, of course, that people are having this struggle. They come in, they get one dose, and it's taken care of. Why did this article say that? Well, because in this population, the data suggested that that was more or less true. So this is data from the Hopkins study, and the red bars are the percentage of people who are in remission, meaning that they no longer, they have a depression symptom score that we consider to be in the realm of normal. And so they dosed it in two different groups, but on average, 65, 70% of people after that single dose were no longer depressed. Now, what's interesting about this, though, look at the time frame, 6 months after that single dose. I saw these data before they were published, and I don't know, maybe I'm dating myself, but if you ever saw Amadeus, Salieri, the first time he sees Mozart's notes and there's no corrections, he realizes Mozart's just writing this stuff down from God, sort of. Salieri sort of drops everything on the floor. I felt like Salieri. I'd never seen anything like this. I was looking at the raw data, and I was like, this is going to change everything. This is incredible. How can a single dose do something like that? And you might think it was an outlier, but then the folks at NYU did a smaller study, but in a very similar population, and found the same phenomenon. They actually followed up in 16 of their original 29 who were still alive 4.5 years later. And different scales give different results, but basically, at least half the people are still in a remitted state. And many said, I had my ups, had my downs, but that experience materially changed the arc of my emotions. Now, how is this possible? We don't know. But I think, as the field's matured, there have been these sort of core questions that have emerged that I think are going to be essential. And one of those core questions is, how do we dose this agent? How often do we dose this agent? But then I want to suggest to you that the answer to that is interlocked with another question, which is, what condition should we be using these agents for? And I want to suggest to you, and I don't have time to make a full defense of this, but I think there's increasing evidence to suggest that some conditions are going to be more amenable to a deeper, more sustained response to psychedelics than others. And I think in particular, in the case of folks with cancer, these were folks that generally did not have chronic depression. They'd been high functioning before developing cancer. And they were upset mostly by a single problem. And I show you here this wonderful paper by Cody Swift called Cancer at the Dinner Table, describing one of the person's sort of hallucinogenic experiences, I think in the NYU trial, where they were sitting with they were having this sort of visualization of sitting with their family at the dinner table, and the door opens and cancer comes in, sits down, and they realize, oh, cancer's part of life. I'm gonna die, we all die. And there was a sort of integration, boom. The person has this different, now this sort of sustained, embedded, heartfelt change in how they look at this problem that has been bedeviling their emotions. And when that happens, it sort of makes sense. If you solve a problem and if the problem is making you miserable, not so surprising perhaps that the effects are long lasting. Now, so we and others, but I can only speak to we, were so inspired by these findings that as Usona started, we decided to pursue an indication. We went to the FDA back now a number of years ago and wanted to do a development program in this sort of condition, and were sent away sort of packing. The FDA did not recognize this as something that they really had a unique code for. And they, wanted to see something, they said, you can study depression. You can study people with cancer, but they have to have major depression. And if it works in that population of majorly depressed people with cancer, we're going to want to see whether it works in major depression writ large. Because, says the FDA, our mandate, our charge is to provide benefit to the largest patient groups possible. And we don't recognize depression and cancer as being any different than depression in general. And at the time, I agreed with that. Roland Griffiths did not agree with that. And he was, I think, right. And so one of my learnings through all this is that many things I thought when I came into this space, I've been humbled and seen and been forced to see things differently. This is one of those. I think Roland was on to the right idea here, that this folks with a particular problem that had generally been high functioning, that was a condition that was especially amenable to this treatment. But of course, once people decide that we're going to try to get FDA approval for something, you've got to study something the FDA will give you approval for. And where do you go? So if you think about depression, major depression, now writ large, the vast cost of the condition is associated with treatment resistance. And treatment resistance is very common. It's not, it's not an all or nothing thing. It's a total spectrum. There are people that are resistant to one drug, and then you put them on another one and they do fine. There are people that are resistant to deep brain stimulation after they failed everything, right? So it's a spectrum. But there has been this emerging consensus over the last decade or so that treatment resistance be defined by a failure of an adequate response to an adequate dose of an antidepressant for an adequate period of time. If you have two of those failures within the current depressive episode, it's generally seen as treatment resistance. And there is a rationale for that. Studies have shown that if you fail one antidepressant, you're not as likely to respond to the next one. If you fail two, you're way less likely to respond. So there is a sort of place where you fall off the chart there. So treatment-resistant depression became a very attractive target for developing psychedelics for FDA approval or for regulatory approval in general. And again, there's a real rationale for it. This is a huge, urgent, unmet clinical need. By definition, they're treatment resistant. They're not responding generally to pharmacologic agents, is what we mean by that. And as I said, they inflict a lot of the high cost of the disease burden. But there's some challenges, of course, also. Despite being fairly widely spread, treatment resistance is a limited patient population compared to the larger universe of people struggling with clinical depression. By definition, it's difficult to treat. And we know that once people get into this space, they don't tend to respond very well to anything. And of course, also by definition, it's less amenable to early disease modification because to be treatment resistant, you've had to have it for a while. So most people with treatment resistant have been depressed for a long time, often chronically, and they've often tried multiple agents, and they'll often be on multiple agents and not getting much of a response. So, but this is, of course, it's sort of the obvious target. And so early studies in the sort of general depression space looked at this condition, looked at TRD. And so there's been the pattern in psychedelic research is academic studies are out there breaking the waves. They're like the icebreaker, cutting open the pathway, and then the big freighters come in after that, these larger FDA-type regulatory studies. So same thing true for TRD here, treatment-resistant depression. And I'm just showing data here from the first open trial in that condition. So there's some things that are important to look at. 19 patients, so this is a small trial. There's no placebo comparator. It's just people with treatment-resistant depression. They're given 2 doses of psilocybin, separated by a week or 2. A 10 milligram dose and then a 25 milligram dose. And these graphs show what happened after that second dose. And what you can see is both graphs show the same thing, which is you see, if you look at the lines on the left, each one's an individual patient. And in general, everybody drops. And they drop rapidly. So this is different than regular antidepressants, right? You get this very, very rapid initial drop. But then over time, over 3 months, you can see that some of the people stay down, meaning that they stay undepressed. But you begin to see this creep up. And then over 6 months, you see the same thing. You see the beginning of this loss of effect. Now, they don't end up back where they started at 6 months. They're still doing better, but they're not doing as well as they were doing at 1 or 2 weeks post-dosing. There's a slow return of the depressive symptoms. And just because we know the folks that did the study, eventually, essentially everybody in this study relapsed. And the other thing to note here is that after 3 months, 6 of the people, so a third went back on their antidepressant, a third received psychotherapy, and about a third, a quarter, or a third actually then went off and did off-protocol psilocybin. So there was a lot of churn there in people trying to hold on to the benefit they'd gotten. And then this brings us to a study now that is rightly famous. The first large regulatorily rigorous study of a psychedelic for a mental disorder. This is the Compass 2B study. It is a study in treatment-resistant depression. And, you know, I'm an academic guy, so I spent years doing academic studies, smaller studies that you do within a university setting. And I've spent the last 6 or 7 years working with Bill and Tura and the Usona team in the world of these really intense regulatory registration type trials. And it has blown my mind, the difference between those two domains. The requirements for these regulatory studies are stunning. I'm like a guy who's had to learn to walk again, trying to deal with the challenges of the rigor that's required for these studies. So studies like this, in addition to having a much larger patient population, are done with a rigor and a thoroughness that is just stunning. And so these data are the kind of data you need for regulatory approval, like with the FDA, and they really are, these are gold standard data, right? So what do we find? So this is a large study, 3 groups, about 70 people in each group, more or less. One group gets a 1 milligram dose of psilocybin, one group gets a 10, and one group gets a 25. The 1 milligram is basically a placebo. And what's interesting here, though, is you see the same pattern that I showed you in Robin's small study 1 slide ago, right? Which is within a day, you see the maximal effect. Kaboom. This is the MADRS, which is a standard scale that is used for registration trials. It's sort of the gold standard clinician rating scale of depressive symptoms, and you can see that at one day the score has dropped in the blue arm, in the 25 milligram arm, by about 15 points. That's a large drop, right? But then what you see is that same pattern that we saw in the small trial, which is there's the beginning of that creep up. And I know Gerd talked about this last night, but I've circled the purple. After 6 weeks, the effect becomes non-significant. Now, if they'd had 150 people in each group, it would have been significant, right? It's just a smaller effect size, and they could have claimed it still worked. These are things that unless you do clinical trials, you don't realize there's these subtleties, and it results in a lot of spin. But what you can see for sure is that over time, the effect is beginning to wane. And then if we look at rates of remission in that trial, remission again meaning that your score is low enough that you're considered to be within normal range. At week 6, the remission rate was 25.3%. You'll see why week 6 is of special interest here. There's two ways of looking at this, right? So one way of looking at this is, wow, we took people who'd failed multiple medications, we gave them a single dose of psilocybin, and 6 weeks later, or 12 weeks later, a quarter of them are still in remission. That is really amazing, right? They weren't taking the medicine every day. Man, that's wild. Or you can look at it and go, wow, I thought this was going to be to a la Matt's talk, I thought this was going to be a cure. I thought this was going to be, 70% of people in remission at 6 months, like the cancer studies. What's gone wrong here? The largest study ever done of an antidepressant was a study called the STAR-D study, where they treated people with Celexa, citalopram, for 12 weeks. And at the end of 12 weeks, they had about a 27 to 30% remission rate. Whoa, this looks like Celexa. That's right. And so there was, those of you that are in the field know that this study was a little bit like STAR-D in my world. It was a bit of a splash of cold water on the face that, oh, this is going to be effective. It's not going to raise people from the dead, let alone maybe save the planet. So that's TRD. We at Usona have a profound interest in MDD, major depressive disorder, and are interested in MDD as a target for a couple of reasons. First off, it's a larger population, obviously, than TRD. Regular old MDD is recognized by the FDA as a condition that you can get a breakthrough therapy designation for. We did, because it's an unmet medical need. And if you look at what happens with a lot of people who are depressed, they have more and more episodes. And as they have more and more episodes, they become more and more resistant to standard treatments. And they also begin to have more and more trouble in their lives, right? That if you're depressed and you can't get out of bed and you're not functioning well at work and life doesn't go your way and relationships are problematic, you begin to dig a hole for yourself that is very hard, A, to get out of, and B, really hard to feel good about. Many people with MDD that have had it for a long time have issues that would depress you too, right? It's just pragmatically, these are very deep holes to dig out of. And so those of us that work in the field have for years, the holy grail has been, can we identify disease-modifying agents? Could we find something that when people come to us with a first episode of depression, we can do something that bumps their life trajectory? So instead of going this way, just bump it a little bit, and it starts heading off day after day after day into in more productive, protective ways. And, so one of the great questions about psychedelics, in my mind at least, for MDD, is if we have them available for the whole spread of the disorder, can we use psychedelics as an alternative to putting people on an SSRI, and then for many people, the years and years and years of taking the SSRI, a la Gerd's, very inspiring talk yesterday. Can we somehow short circuit that? Because, one of the things that's increasingly apparent is that long-term use of antidepressants appears to make people dependent on long-term use of antidepressants, partly for relapse, and then partly perhaps because the brain changes. When you have a drug on your brain all the time, the brain adjusts. And, what's very clear is that when you've been on an antidepressant for a long time and you stop it, you're at much greater risk of relapse. So let's talk about MDD. So this is sort of the equivalent of the study I showed you, the small open trial, showing that here in MDD, 2 doses produce a large antidepressant effect that is sustained out to 4 weeks. This is a study from Hopkins. They then gave everybody the psilocybin and actually followed people out for a year. And look at those response and remission rates. They're looking a lot better. Now, this is a small trial, but again, raising the possibility that maybe MDD looks a little bit more like those cancer patients. There were 2 additional, slightly larger academic studies. The study on the left is the study that was done at Imperial, comparing Lexapro or escitalopram to psilocybin. And the headline was that on the primary endpoint, the 2 were equivalent. If you look at the MADRS, which is a scale I'm going to show you in our study, you see that they actually had about a 14-point drop in the MADRS. That looks similar to what the Compass study found. And they had a 7-point drop with their escitalopram, which interestingly is about what the Compass study saw with their 1 milligram placebo. And as I'll show you what we saw. And then over on the side there, I'm going the wrong way. Is there a way to go? Is there a way to go back? Oh, wait a minute, sorry about that. There it is. And then the other study, a smaller study from Switzerland, from Franz Wollenweider's group, showing the same thing. You give people in this case, a 15 milligram on average dose, which is lower than the 25 milligram that the Compass study used or that we used, but you see the same rapid drop. And the final endpoint was only 2 weeks out, so it doesn't provide information on longer endpoints. So, as the Compass study was the sort of first large registration type trial in TRD, I want to show you now a study that was published yesterday online in JAMA from Usona, looking at the same 25 milligrams single dose psilocybin. And the first thing I want to say is that, man, it takes a village to do this. Look at all these authors. They're leading people from 11 institutions around the United States, and there were so many more people that were involved in this. I had never been in a study like this where I was just such a small sort of cog in this large, amazingly well-organized wheel. And so shout out to this amazing group of people that have made this possible. Here's the study design. This looks like a crazy rat's nest, but it's really a fairly straightforward study. We took people with major depression. We gave them 6 to 8 hours of prep. We gave them a 25 milligram dose of either a 25 milligram dose of psilocybin or a 100 milligram dose of niacin, which is a sort of active comparator. And then they had their dosing session, and then we followed them for 6 weeks. We did a 2-hour integration session the morning after, and then an additional hour at the weeks after that. We had a number of outcome measures. 6 weeks is the primary endpoint. The MADRS scale is the measure of depression. What did we find? Well, before that, we had, at the end of the day, we had 51 people in the psilocybin group, 53 in the niacin group. We lost a few more people in the niacin group than the psilocybin group because people get frustrated for all the reasons of blinding we talked about. There's a little bit of a higher dropout rate. The average population, this was evenly divided between men and women. These were middle-aged folks on average. They were overwhelmingly white. We could talk about that. That is a challenge for the field. But this is interesting. Only 13% of them had treatment-resistant depression, and that's going to be important. 30% in the psilocybin group, 15% in the niacin group had to undergo a medication taper. So most of these people were not on an antidepressant, which is also interesting. But their baseline score of 35, that's the beginning of very severe depression. So this is a severely depressed group. They've been depressed for a while, but many of them are not taking an antidepressant, and most of them are not treatment resistant by the standard definition. What did we find? Well, here's the niacin, here's the psilocybin. And what you see here is that psilocybin produced a large, rapid, I don't have time to explain it now because I'm going to run out of time, but you can ask me a question about the day 2 data, a very powerful drop in depressive symptom score. The score dropped 19 points, as opposed to, say, 14 in Robin's study against escitalopram and about 14 in the Compass trial. So there's a larger raw drop from baseline. And the other thing that's striking about this figure is you don't see the line going back up again. Now, we don't know what would have happened after 6 weeks, but out to 6 weeks, in fact, the effect was numerically larger at 6 weeks than it was at 1 week. So in this population, the effect was large and it was sustained over that time period. I love this graph. They forced us to do it. I'd never heard of it. They called it a waterfall or a spaghetti graph. I'm going to take a second to explain it to you. Each orange line is a patient that got psilocybin. Each blue line is a patient that got the niacin. The baseline is going up like a mountain. So up on top are people that were just depressed as all heck, almost 50 on the MADRS. How far they drop is how well they did. So the gestalt, you just look at all the lines, you say, oh, wow, yeah, there's definitely more orange lines that dropped a long way. And many of the people that were most depressed dropped the furthest. The blue folks, yeah, no, not so many of those guys are dropping. But there are some that had a full cure. You see that? It's amazing. They got the placebo. But then look, there's more blue lines that go up, meaning they got more depressed. But there's a couple of few orange lines that went up too. Not everybody benefited from this. That is a really important point. These are the raw data, and I will never do a study that I don't show this sort of plot again, because it really gives you a sense of it that is more in depth than the standard line graph. What about response and remission? Our response rates at day 43 or 6 weeks were 60%. Our remission rate was 44%, again, compared to, say, 25% in the TRD population. So a much higher remission rate. There is a scale called the Sheehan Disability Scale. It just measures how well you're doing in your life. We saw a big improvement there compared to the niacin. And then there's a bunch of other measures. So clinical global impression is just a measure of how overall impacted you are by the disease, how severe is the disease. That improved significantly. People were less anxious. They had better quality of life. And then I want to take a sec to talk about this. We had a self-report depression measure, and it allowed us to do something interesting. So the baseline for this study was about 5 to 7 days before the dosing. During those 5 to 7 days, people got to prep sessions, right? So we had this long discussion early on in this. When do you count your baseline? Should the baseline be before the prep session, so you consider the preparation as part of the treatment? Or should we put the baseline right before the treatment, have the prep sessions out here? So for the MADRS, for the primary endpoint, we decided that the prep sessions were part of the treatment. We start the clock ticking here. But with this scale, we were able to measure it here, after the prep, right before dosing, right? And then later. And so what this shows is, if you look at the drop from baseline in this self-report measure, it's twice as big in the psilocybin as the niacin group. But then if you start it now, if you look from just before dosing, how much of that effect was explained by the preparation? The good news is not very much. So just if you look at the psilocybin, you can see that if you go from baseline, the score drops by 21 points. If you go from right before dosing, the score drops by 17 points. You see the same kind of adjustment in the placebo, suggesting that, yeah, the prep seemed to lower the score a little bit, but the vast bulk of the work was done by the medication or by the medication dosing session. If you want to be more specific, we did not see any blunting, which is important because, as Gerd was talking about yesterday, standard agents really do that. Now, remember I said only 13% of the people had treatment-resistant depression. So we did a little sub-analysis where we looked with the caveat that that's a very low number, so we don't have much statistical power. But take a look at the people that did yes versus did not no have treatment-resistant depression. If you had treatment-resistant depression, your score dropped by 14.5 points. If you did not have treatment depression, your score dropped by almost 20 points. Interestingly, that 14.5 drop in the MADRS is almost exactly what they saw in the COMPASS trial in TRD. By week 6, theirs was more like 10 or 11, but their maximal drop was right about there. So it's pretty interesting. The p-value over here on the far right is not significant at day 43, 6 weeks. But I will tell you that if you looked at the earlier time points, even though the numbers were small, it was already statistically significant. The people with TRD did not have as large a response. And so this raises to me now, I think, one of the key points in this field that I've sort of organized this talk around, which is the condition that you address the drug for is going to influence the effectiveness of the drug. And it seems to me that there's sort of a gradation of probably conditions that are amenable to these sort of powerful, powerful responses. Probably things like cancer-related distress at the top of that, something like MDD in the middle zone, and then TRD, because by definition it's more difficult to treat, of benefit, but not as large a benefit as MDD writ large. What about the actual psychedelic experience and its effect on outcomes? People that got psilocybin had much higher scores afterwards, after the dosing day on the mystical experience questionnaire. They had much higher scores on the emotional breakthrough inventory. We did a path analysis, and interestingly, in this population, Yeah, only the MEQ predicted outcomes, but it predicted it powerfully. So what the fancy graph on the right says is that we looked at that there was about a 14-point difference between psilocybin and niacin at the time point where that was maximal, which was at 4 weeks. Of the 14 points that by which the psilocybin beat the niacin, 11 of those points were accounted for by the MEQ. That's a huge effect, right? So again, this is very strong evidence that the psychological, experiential thing that happens during the dosing session is, in ways we do not understand, connected with the mechanism of action. The adverse events were generally mild: headache, nausea, visual perceptual effects were the main ones. A couple of people reported ongoing sort of mild visual effects for a couple of days afterwards. There were no serious treatment emergent events. Give me just a second, I'm almost done. So then what's next? We are in the midst of launching a phase 3 program. We've been exploring global regulatory pathways, and we have built this astounding building that is completely dedicated to the science and treatment and clinical use of psychedelics. Let me go back here. This is a 10,000 square meter building. It's going to be open in 5 minutes, 10 minutes. How long, Bill? We're right about there, right? It's going to be open this fall. It's going to be a place of convening, as I say, of treatment, of research. It's really going to be a unique resource, I think, for the world for psychedelics. And then finally, here are the QR codes for both the article and a podcast that they published contemporaneously with the article. And we've had so many collaborators that we can't thank them all. But I can thank you. And if we have time, we'll take some questions.
So this is really exciting, and as you may notice, Chuck had more time to speak because we can run into the break and use the break for discussion. This is exciting data, and I'm sure that you are coming up with some exciting questions. So who wants to go first? Up there.
Yeah, hold on. I'm going to go back to the QR code. Good point. There you go.
So my question would be, what is it then about the treatment-resistant depression that makes psilocybin treatment less effective?
Yeah, that's a great question. We don't know, obviously, but I mean, if I was to theorize, I've become increasingly concerned that our standard antidepressants, when used chronically, can begin to change how the brain functions. There really is some evidence for that, right? So, basically anything that sits on the brain chronically and affects the brain function, thereby affecting its sort of microstructure, is going to change the brain. And, we know that antidepressants make people less responsive to antidepressants. Many people, not everybody, but many people. And I suspect it's the same thing, because I think it's pretty clear that psychedelics, their immediate biological mechanism of action is through serotonergic signaling. And antidepressants generally that are used in the West modulate serotonin signaling. Now, what we don't know, for instance, though, is whether if somebody had done nothing but take Wellbutrin for years, and if they failed Wellbutrin, they went to reboxetine. These are agents that are noradrenergic. Would you see that same diminished response? That's a study somebody should do, because one prediction might be that no, you don't see as much of a diminished response because you haven't spent years, with this sort of chronic manipulation of serotonin pathways.
So thank you for the talk. I had a question specifically about the path analysis that was used and the selection of the MEQ as the psychometric tool to measure the phenomenology of altered states, because there are two other ones, which is Pacala's PCI and Dietrich's 5-DASC. Now, the MEQ kind of developed out of this idea of this intangible kind of relationship between religion and altered states, whereas the PCI and the 5-DASC kind of came from this philosophy that all altered states kind of share this invariant dimension. So one, I wanted to ask, why the selection of the MEQ? And secondly, do you think you would see similar results if you use the PCI or the 5DASC?
So the selection of MEQ, I think, was historical. We were working closely with the folks at Hopkins. It had begun to show these associations. It really is not a defense past that. The EBI, because as we were putting the study together, I was very cognizant that the MEQ was tapping into one sort of domain that these agents seem to produce, these heightened kind of expansive states. But we were seeing so many people that were also sort of struggling where these agents seemed to be producing cathartic, like psychologically based things. I think we would see the same thing with the 5ASC, the other instruments that tap into some of these same constructs. For instance, oceanic boundlessness, you'd probably see the same thing. Obviously we didn't administer those scales, but it's pretty consistent. I mean, Robin saw with the 5ASC, same thing, that there was a little series of questions that pretty strongly predicted outcome. So I would expect you to see the same thing. Although Franz, the Swiss study, the 5ASC, it did not, I don't think it did correlate with outcomes, did it? Yep.
With the psilocybin group, but it was depending on the placebo group because we had some responders in the placebo group too, very strong. But we had to decompose to separate out what's more disembodiment versus emotional modification, because the oceanic boundlessness includes increase in emotion. And we saw that emotions drive a lot the change in after 2 weeks or 3 months, and even it was replicated in a longitudinal fMRI study, which we haven't published the data yet, where we see that psilocybin alters the processing of emotion. It's not so much increasing emotion. That's not so interesting for us. We like to understand more the mechanism behind it. Is it cognitive alterations like a loss of ego boundaries in terms of disembodiment, or we use more sophisticated neurocognitive approaches to that, like ownership or body experience using predictive coding. We'll talk about that on Saturday. So we try to crunch down this huge dimension of oceanic boundlessness, which is too broad to have as a mechanistic understanding what's driving the change. So I'm not really happy. We do it, certainly. It's correlation, it's not causal, it's no explanation. We have to be very cautious about that. I don't believe in these correlations, even if we do it a lot. So it gives a good hypothesis where we have to go deeper.
Absolutely.
What I could add is, in the study you showed, the slide, it's still down after 3 months. There's a response rate of about 45%.
No, the study was 6 weeks. So it's at 6 weeks, it's 45%. Yeah, we don't know past that in the population. That's a limitation, obviously, of this study.
Yes, hi. You mentioned earlier that the depression symptoms are only part of the picture and the life circumstances play an important role. And ideally, we would like the treatment to also assist people in making changes in their life that lead to lasting outcomes. Can you comment in some form on how psychedelic treatment compares to classic SSRI or something else in terms of supporting making life changes?
So that's interesting. I don't know that, the study that would address that would be the Imperial College study against escitalopram, right? And there's a whole bunch of literature coming out of that. David Eritzo's here, I don't know if you'd know, but there's some evidence that the psilocybin in that study may have done things that would be beneficial for making those sorts of changes. So for instance, the psilocybin produced much higher rates of feelings of long-term social connectedness compared to the SSRI. But it'd be really interesting to do a study sometime and look and see, when the psilocybin works, do they actually become more engaged in their community? Do they actually begin to exercise? Do they actually, ... We're working on a study out in Colorado where we're going to actually try to, through UW, that we're going to try to look at that, because it's such a key question, right? Because, you know, if you have a profound experience, but you go back into a social circumstance or lifestyle that is depressogenic, you know, you're paddling upstream, right? It's not surprising that you're going to lose the effect. So this is the question of how do we optimize the endurance of the psychological benefit from a psychedelic is, to my mind, maybe the most important question in the field, you know? And it's either going to be optimized by repeat dosing, probably, right? Maybe sort of a pharmacologic model. Personally, I'm very interested in the question of, can we find ways to extend the benefit without having to do a lot of repeat dosing? And it touches upon what you're saying. But, you know, studies haven't looked at that yet. But it's a key question.
Thank you so much for this talk, Charles. So I was wondering about the center that you're building. Can that affect the results, do you think? That you go to this beautiful center and you get this psilocybin experience and it works synergetically?
I would guess yes, because every time I go in it, my depression score drops fairly significantly. It has an amazing sort of downstairs area with steam rooms, and I also study heat and stuff like that. So this is like, you know, kind of this dream place for me. That would be a really interesting question. You know, a lot of things will be done there. I think there'll be mostly treatment and educational type stuff. But that would be, you know, these are all fascinating questions, right? You know, I think Robin Card-Harris is at UCSF, not with our building, but is trying to get at this, where he's started a study where he's manipulating not the psychological set, but the way the room looks, right? And so, yes, I would think that if you were dosed in the new Usona building, that you would get added benefit. The setting is, you know, it's a Mondo setting. I mean, it's really amazing. Yeah, good question.
So we can take one or two more. Yes. Let's make this more sportive.
Thank you. Why do the studies stop collecting data after 6 weeks? Because it would be even more interesting to see what happens after 6 months or even 6 years.
Infinitely more interesting. And every study going forward now is going to collect data for a year at least, right? Because remember my first slide, right? You know, if you give a group of people that are depressed and anxious a single dose of psilocybin, and the vast bulk of them are fine for months and months and months afterwards, that leads to a different way of conceptualizing how the drug would be used clinically than if, say, people on average needed to be redosed every 2 or 3 months, right? These are very, very different paradigms. So everybody in their interactions with the FDA in the last year or so, it's just become there are several things that become expectations moving forward. One is that you follow people for a longer period of time. You know, the FDA generally will accept a primary endpoint for a depression study somewhere between 6 and 12 weeks. And of course, you say, well, God, depression's a chronic condition. Why have all these studies been 6-week-long studies? Well, the assumption is that, you know, if you're taking a pill every day and you're doing well at 6 weeks, you're going to be doing well at 6 months because you're still taking the pill. By the way, not true. You know, lots of people get tachyphylaxis, but that's the fantasy, right? But it's a different scenario when you're taking a pill and then it's gone, and then you get this benefit. You know, you think, well, how long is it going to last, right? So determining redosing of the need for redosing and scheduling redosing is going to be a major push of all development companies, all development programs working with these agents. And then another push that's related to that we've been talking about is blinding. You know, that people are going to want to know, you know, how effective was the blinding? And the answer is going to turn out that the blinding was not particularly effective. But even with that point, you know, it's so interesting that, you know, if you look at the placebo response in the Compass 2B study and the Franz's study and our study, you know, whether it's 1 milligram of psilocybin or an inert thing or niacin, you see about a 6 or 7 point drop in the MADRS. That's actually what Robin saw with escitalopram. So it seems like there is this replicable, modest improvement that occurs in placebo conditions in these disorders, and we assume even though people realize they they're unblinded. So blinding and placebo response are not one-on-one on top of each other. But you're right, and I digress. Studies going forward will follow people for at least a year.
Very encouraging data. So if you could redesign, that's the last one, redesign an element of the study or two, now after having done it, where would you start to optimize the design?
The first thing I would do, I'd do an endpoint further out. It's hard. It's hard to do a longer out endpoint, like 12 weeks, because the people that got the placebo, you know, who because of the unblinding generally know they got the placebo, we're asking them to stay off antidepressants. So different from the COMPASS trial where people could go back on an antidepressant after 3 weeks, we kept people off, right? And there were a couple people in the placebo group that couldn't take it, that, you know, they went back on an antidepressant. So it's a challenge because you really want to know, I don't want any other treatment in that 12-week period. But I do think it would be interesting to have a longer follow-up that way. You know, again, what we were just talking about, I think a robust year-long follow-up to see how often redosing needs to happen. I mean, all studies will do this. I might have administered, to that gentleman's question, I might have administered another scale or two, trying to assess the immediate psychedelic experience to better understand what is the phenomenology that associates with outcome. And then, of course, what's really become of interest to me is this question of if you take people with MDD that are not treatment resistant, how much of a benefit differentially do you get? And is it enough of a benefit to support the use of MDD for payers earlier in the disease course? Because I'm personally convinced that these agents are going to be most effective being used earlier when people are struggling with the first depressive episode. So that, you know, in my vision of the world of the future, what I'd love to see is that people that come in, you know, that are younger, that are beginning to struggle with depression, have sort of an option. You know, you sort of have the standard option where you can go on standard antidepressants, or you have a psychedelic option where you try that, and you may still end up on standard antidepressants down the road, but I would predict that some percentage of people would be able to then not enter that chronic antidepressant use if they got a psilocybin treatment earlier. And so yeah, we'd really like to know more head-to-head about TRD versus non-TRD depression.
Thank you very much, Chuck, Raison, and Usona for presenting the data. That was a good start into the conference, and so have a good rest of the day. Thank you.
Thank you.